Leong, ela já vem colaborando com a gente desde o começo do nosso projeto Germina, que alguns conhecem como Brainrise. Ela é professora titular de Developmental Cognitive Neuroscience e diretora do Early Mental Potential and Wellbeing Research Center na NTU, que é uma universidade em Singapura. E desde o começo, né, do Germina, do Brainrise, ela trabalha com a gente, ela faz parte do 1KD Program, que é o programa que o Brainrise faz parte. E a Vicky, ela está muito preocupada em entender a neurobiologia da interação dos pais com a criança. Tem feito, ela é pioneira no que a gente chama de EG diádico. Então, na maior parte das pesquisas que a gente tem em psiquiatria, medicina de maneira geral, a gente tem sempre interesse no indivíduo, e ela vai além disso, né, preocupada então com a interação social. Então, é uma unidade, uma dimensão diferente do que a gente está acostumado, né? A gente coleta indivíduos e aí a gente vê se TDAH, se depressão, se esquizofrenia está associado a alguma variável, se tem diferença entre grupos. Ela tem interesse nessa DIRD, né? nessa interação. Então, sem mais delongas, eu vou convidar ela para falar um pouco da linha de pesquisa dela nos últimos anos. Acho que vai ser bem interessante. Depois a gente vai ter tempo para perguntas também. So, I think that's it. So, the floor is yours. The microphone is yours. Let me just...
So thank you very much, Daniel, for that introduction. I didn't completely understand it, but hopefully it was positive. Thank you again for the invitation to visit you again in Sao Paulo. It's a real pleasure to be back in this city. I was here about last December before this, yes, for my very first visit. That was to set up a collaboration with the Germina team, and that has been incredibly, fruitful, and definitely is something that's been triggering a lot of my thinking about cross-cultural issues and how we can think about development in different cultural contexts. So it's something I hope to continue in the future. So today's talk, I wanted to give you more of a personal insight into my journey as a researcher. I have chosen for my career path quite an unusual topic. So I've chosen to work pretty much at the edge of what people would consider, acceptable in some senses. When I talk about neurosynchrony, many people worry that I'm talking about telepathy, but that's definitely not the case. We're working with a phenomena where people are able to understand and interpret each other's social behavior, and with that comes some kind of alignment in our brain processes. But explaining that and walking that very fine line between what is really aspirational and discovery and what people accept has not always been an easy journey. So since some of you, or maybe most of you here, are scholars yourself, I thought it might be helpful for me to share some of my learning and some of my struggles even in understanding and breaking new ground in a very novel field like this. So the title of my talk is Paradigm for Neurosynchrony. And two things which I still, am balancing in my head. The mechanisms. How does this phenomena that we've been able to measure come to be? What are the processes that support synchrony that we are able to measure? And secondly, what are the questions we still can't answer? What are the mysteries still that people maybe like yourself could take on as questions in the future? So just to get started, what is synchrony? So synchrony is a state of contingency where the behavior or the physiology of one partner is predictable with respect to another partner. So there is some kind of relationship or contingency between one set of temporal signals and another set of temporal signals. It doesn't have to be as simple as we're imitating each other, so we do the same thing at the same time. That's the most restricted interpretation for synchrony. And when I say synchrony, it means there is some kind of an association or a dependency of one partner and the other. And the best way I can describe it is something like dancing. So if we see a very well-rehearsed pair of like tango dancers, for example, they're clearly not doing the same thing at the same time. If they did the same thing at the same time, that would be like a marching in a parade, right? That would not be dance. A good pair of dancers is attuned to each other's movements. So they are moving, but they are moving with the same tempo and the same rhythm, but they're doing different things almost all the time. And they're anticipating and responding to each other, but there's a common temporal backbone which is guiding their behavior. And that's what I mean by synchrony. There's contingency, there's an overarching tempo that's coordinating their behavior, but it's not as simple as same thing, same time. Okay? So actually, when we interact with other people, we achieve synchrony to greater and lesser extents. And you can feel this in your gut sometimes. When you talk to some people, they're kind of getting you, they're kind of responding to you, they're not interrupting you, you're building knowledge together in a very smooth, cumulative way. And for some other people, it's just really jarring. It's like, okay, we're not really on the same tempo or the same wavelength here. So you can sense that. And interestingly, we can start to measure that with techniques these days. So in the lab, we've looked at these synchrony states, and we're particularly interested in synchrony between the parent and the child, because during that very early period in life, developmental psychologists have noted that there is this very closely attuned or dyadic dance that the parent and child engage in. The other thing I want to highlight about synchrony is that it's quite a transient state. It's not the case that once we're in synchrony, we stay in synchrony for the next 2 hours, right? It's a really transient state. You couple, decouple, couple, decouple. That means there are events in your environment which cause you to align. Oh, okay, suddenly we're in the same rhythm. And then after a while, that rhythm dissipates. So it's a transient state, and we can study its transience as well and the cues that bring you in and out of synchrony. So we've measured the context and the cues which seem to promote synchrony, things like eye contact, changes in the way that we speak to people which raise attention or which indicate salience or intentionality. And emotion seems to be a very powerful cue that can synchronize people. So social context and social behavior are really important to understand what brings people together in synchrony. And we're also interested in the functional consequences. So is this just some cool thing that we can do and it's not really meaningful? Or is it something that's quite fundamental to our ability to coordinate and work together as a socially orientated species? And actually, I am obviously leaning towards the latter interpretation, that, being a social species, we have to learn to work together and coordinate our behavior. Hence, the ability to achieve some kind of coordination and synchrony is important. And of course, that varies as a function of the type of relationship that we're talking about here. So other researchers have talked about...
I think we are not sharing the slides, but sorry.
Oh, sure. Oh, not on Zoom.
Yeah. My bad.
No worries.
Tá certo? I think
we're getting there?
Yeah.
Okay.
Sorry.
Cool. No worries. Okay. So we were talking about. Is that okay?
Yeah. No, it's okay.
Super. Okay. So other researchers, particularly people like Ruth Feldman, etc., have been interested in the effects of synchrony on social bonding and social affiliation, particularly through the oxytocin system. And I think that's a really crucial angle to look at, because I think high levels of contingency and synchrony do seem to cement themselves in the longer term into certain attachment styles or stable affiliative bonding. But because I'm a cognitive psychologist and neuroscientist by training, I'm really interested in how it boosts or focuses our cognition and changes our behavior and our strategies for interacting with the world. So a lot of the experiments that I've conducted have looked at synchrony in relationship to communication, learning, and indeed decision-making and exploration in the world around us. So how do we measure synchrony? I think some of you have come across this already in my papers. We do dyadic EEG, which is concurrent collection of brain signals from 2 partners. Usually it's mother and baby, but it can be any other pair as well. And then we use modeling methods like range causality to pick out directional influences of one partner on the other. And so I started looking at these social signals like gaze and speech because of a theory in psychology called natural pedagogy. And this assumes that as a human species, we've developed and we fine-tune like a code for communicating information that is relevant for learning. And we do this particularly for young members of our species like babies. Because babies are in a very busy, buzzing world where there's a lot of information in their environment. And one of the things that caregivers have to do is help them focus their attention on information that's relevant for learning so they can focus on learning and ignore everything else that's irrelevant. So the idea is that human species have certain signals that we use to tell the child that this information is relevant for you. And these are called ostensive signals. And in fact, almost all the things I talked about in the previous slide fall under this category. These are things like eye contact, using the child's name, and using infant-directed speech. And they have certain properties. They're very perceptually salient and discriminable. And if you use them, the baby usually turns their head to orientate towards the source. We know that in the context of development, if the parents don't use these cues consistently, for example, if the mother has chronic depression, they have less contingent eye contact, more flattened speech, then this has links to the child's ability to acquire linguistic information and to tie that to events in the world. So language development in particular seems to be compromised if the parent doesn't use these ostensive signals very effectively in their interaction. So I started studying these ostensive signals and their effects on the child's brain with the idea that maybe this might be one of the ways that we synchronize the partners that we interact with. So my early work was looking at child-directed speech. And without going into too much detail, you can read the papers if you want. We found something quite interesting in the structure of child-directed speech. Now, when you hear it, you're straight away struck by the melodic changes. It's hello, how are you? It's really colorful, expanded, the melodic range is higher, it's normally slower, more emphasized. But actually, if you look at the rhythm patterns, there's something else intrinsic in that too. It's much more regular. And the stress patterns are also more regular. And one of the things that helps the child to do is segment and parse the speech signal into the onsets of syllables and the onsets of words. So this is interesting. So I developed a computational model which modeled these rhythm patterns and showed that you can indeed do speech segmentation using these rhythm patterns. But going beyond that, we looked at what the actual rhythms were, and we found, to my surprise, that they were very perfectly matched to the endogenous oscillation rates that you can measure using an EEG signal. So all of us at rest have certain canonical EEG frequencies at which your brain is naturally oscillating and which you're able to receive and parse information. We call these the delta, theta, and alpha bands. Those are some of the most prominent ones you can measure. And in fact, in child-directed speech, there are tiers of information that are packaged to match these three rates almost perfectly. The stress patterns come at the delta rate, the syllable patterns come at the theta rate, and the onsets, so the breaking part of the initial consonant and the later vowel, that matches the alpha rate pretty nicely. So it's quite nice that there's this serendipitous match in the temporal structure of how we speak to children and what their brains are prepared to receive, which tells us that maybe there is this biologically privileged system that allows us to speak in a way that entrains the oscillations of the child listener to help them break into speech and parse it really easily. So this was my first foray into this idea of entrainment and synchronization. I then started to look at other cues, and this is probably the most famous paper that we put out looking at eye contact. And we found that when the adult speaker was looking at the child while singing to them, compared to looking away, her ability to synchronize the child's brainwaves was significantly higher. So you can see that both in the alpha band and the theta band, whether you were looking at the influence of the adult on the infant, or the other way around, the infant on the adult, eye contact was a powerful synchronizing signal that raised the brain-to-brain synchrony between the adult and the child. Now, during this experiment, the adult could also call the child's name because sometimes the child was bored and distracted. So she called the child's name and said, Tom, look here. Or she waved her apple. And we could measure brain-to-brain synchronization before and after the cue. And we found that this vocal cue was also effective in increasing synchrony temporarily between the dyad. And just to show you that it's not just the adult who can synchronize the child's brain, we also measured the child's utterances. Because these were nursery rhymes that the adult was singing, like the wheels on the bus go round and round, very familiar ones, the children got quite excited and some of them started babbling and cooing along to it, so we could measure the vocalizations that the children were giving. And we found, so each of these dots here is one kid. The children who vocalized more, made more of an effort to communicate, they had a stronger synchronizing effect on the adult's brain. So what we're seeing is this multimodal reinforcing cycle. The adult kicks things off by looking at the child to specify their intention while they're singing. That synchronizes the child's brain. The child then releases their communications, which back synchronizes the adult's brain, and the whole thing gets sustained. It's this nice positive feedback cycle. And we see this in this nice instance where you have a very rhythmic frame because the adult was singing in nursery rhymes. So you have this overarching temporal structure, which is binding everything together. So in this context, we could demonstrate that there are multimodal social cues that can raise synchrony and that both partners have an active role in sustaining that. So that was quite nice, I thought. It also motivates why you should sing to your kid, in case you don't already do that. We then explored other social cues, like affective cues, and people here who are studying mood disorders might find this interesting. So we had another paradigm where we asked the mother to display positive emotion or negative emotion when describing objects on the table to the child. And we very simply measured the brain-to-brain connection between the mother and child during her expression of positive, really bright, colorful, happy affect. And negative, almost disgusted looks on her face when she's like, I want to avoid this, I don't like this, those kinds of facial expressions. And to our surprise, this was a really powerful effect size. I mean, I'll show you later the effect size of the gaze, but this was several times the effect size of the gaze effect. The emotion was a really powerful driver of the integration of the brain networks between the mother and the child. And so here we also did some methods development for the field. As you know, graph network theory is one popular way to calculate connectivity within individual brains. Here we adapted the metrics for cross-brain indices. So we developed a new measure called divisibility, which previously was only used within brains, to measure how closely networked together two brains are. Or simply put, do they function as one big unit or two separate units? And we found that during positive emotion, there is this great integration into one mega unit. So divisibility was very low during positive emotion. During negative emotion, the two brains segregated out like that. So one of the great ways, if you want to have someone aligned with you, is to show positive affect, well, at least with babies. And the other nice thing that we thought, because we wanted to make sure it wasn't a fickle effect, we did two types of measurements, a directed measure like PDC that I introduced to you, and a more standard non-directed measure like phase locking value. And so they're based on different parts of the signal. One is phase, one is amplitude and phase, but across both of them and across multiple measures, we saw this replicability in this effect. So actually, for my money, the emotional power to synchronize is actually more in this domain than the other cues. The next one is probably the auditory cues are the next most powerful. Okay, but is this all? Because I've given you the picture that it's all about the behavior and the cues that we're giving, and so if you can somehow train yourself to generate the right cues, you'd be synchronizing everybody around you, right? It's not as simple as that. It's not just bottom up. Because in the gaze experiment, we had a third condition. So I showed you about the direct, looking straight, indirect to the side, but we had a control condition, which I call the Mona Lisa gaze condition. So this is where we want to control for the side face view, because this is less familiar for babies, right? So we wanted to make sure she was looking to the front, but her face was to the side. So we dissociate the gaze from the face, okay? And we expected that if the effect was completely about the eye contact, we should get the same level of synchrony for direct versus Mona Lisa gaze, because in both cases, she's looking to the front and the side face doesn't matter. To our surprise, the synchrony in this third condition was the highest. It was not the same, it was the highest. It beat even the direct face view. So this tells me that, yes, gaze is important, but gaze isn't the whole story here. There is a bottom-up effect, but there's something else top-down that's telling the child, pay more attention, or this is more relevant even than in the direct gaze condition. So we know that it's not just as simple as the type of cue. It's not just the bottom-up behavior. It's also the interpretation, the ostensive value, or the salience value of the signal that also matters. And with that comes other things like, ooh, does background history matter? And all the other questions. So it's not a simple picture, but we're starting to see bits of that puzzle. Okay, so here's where we get into, really, this is a pretty picture that I presented to you, but does this actually work? And one of the biggest challenges we've been struggling with, and I don't think we've solved it completely yet, is can we see this in the real world, in naturalistic conditions? Because I showed you an experiment which was very highly controlled. We crafted it to elicit maximum synchrony there. All singing, all dancing, you know, very controlled angles, and everything was really very well tuned. In the real world, obviously, we don't have situations which are that controlled. And one of the biggest problems for us in EEG measurement is movement artifact. Those who work with our EEG signal will know this. In the case where the baby is fairly attentive and sitting and looking at the screen, like instructed paradigms, which was the PNAS paper, if you hand code the proportion of time a baby's moving anything at all, and yes, I make my graduate students do this, they watch the video and code when the baby's moving anything at all, then somewhere between 60 to 70% of the time, the baby's moving something. And that means they're generating movement artifact. So we've got to worry about what to do with that. In our PNAS paper, we discarded all of that data. So we were left with less than half the data set. But that was to make sure we were very confident in the cleanliness of the signal. Now, if you want to go into the real world where they're enjoying and playing and doing fun stuff, what happens to movement? Well, we hand-coded this in another paper. If mom and baby are playing, obviously they're moving over 95% of the time. So if you were to do the same thing, just reject the data, you have zero data left. So clearly, it's not a solution to discard data where there's movement. We have to have better solutions for this. And to be honest, the field is not there yet. There isn't a good solution for this. Either you have to live with some motion artifact in there, or you have to discard 95% of the data. Neither is a good solution. So let me show you why this matters. If I go back into my gaze paradigm that I showed you with that nice effect of brain-to-brain synchrony, I told you that we scrubbed out all the movement, right? What happens if I put it back in? So here's a simulation where I put back, oh, it's just painful, put back noise into the signal, just a little bit at a time, 1%, 3%, 5%, 10%. And as you can see, even adding 1% of the noise back in, and our effect disappears. So this gaze effect, it's there, but it's only there if you can control for every other contamination that is possible to be present. And obviously in a real life interaction, you're not going to do that very well. So it's a good question is whether you can actually observe the same effect in the real world with the methodological constraints. Maybe emotion is a bit more robust, but this one I think we will struggle with in the real world. So how are we trying to get around this? Well, hopefully there is machine learning and computational methods to the rescue. Some of you have started hopefully familiar with things like ICA, which is an artifact removal technique. But a lot of that depends on you hand annotating signals, and it depends on a blind source separation algorithm. So you don't really know whether we're taking out brain or movement. So what we're trying to do is train a machine learning model to recognize movement. And we do that by using the movement patterns of the mom and the child. So the Davina team will know that we've been working with them on pose estimation in the PCI signals. So this is where we can just take a regular video of a mommy and baby interacting and superimpose the joint movement patterns of 25 key points at every single video frame. That gives us a real-time estimation of what is moving and when. So what we're able to do with this then is to use those trajectories to train a diffusion model and then predict what it's doing to the EEG signal. And then with that informed, kind of intelligent way of removing the artifacts, maybe we can do a better job of cleaning out the problems in the signal. So we have some good preliminary evidence that this could work. We've been able to do a saliency map validation of this. And compared to some state-of-the-art models that are already out there, we're already doing better in terms of our RMSE. So this is potentially in the works, but there's still a lot more tech development that we need to do before this will be usable for the general cases. So in the second half of my talk, I want to tell you a little bit more about what I think is going on beneath the surface. So we've been measuring at the top these phenomena, and I've been telling you a bit about the social context, but what's driving all of this. So to get underneath the surface, I think, unfortunately, we can't use the babies because you can't be invasive with the babies and you can't really put them through the rigor and the number of trials and the amount of data that you need to really model things robustly. So for mechanisms, I've turned to two other models, adults and animals. And although they're not the same, as adults we're very different in the way that we behave compared to babies, but at least we can get some more robust measures of the underpinning mechanisms. So bear with me. This is a bit geeky, but it's my constant rant about how limited we are in our understanding of what synchrony is. So I started by saying that synchrony isn't about doing the same thing at the same time. And actually, that's another way of saying that we've only been thinking about this as a linear relationship. A changes with B in a linear way. A goes up, B goes up, or A goes up, B goes down. But very rarely in biology do we get that type of simplistic relationship. Very frequently, we have non-linear relationships, where the distribution of one predicts the distribution of the other, but in a completely complicated way. Right? So most of the measures we've been dealing with are linear. These oscillation-based measures like PLI, GPDC, they assume some kind of up goes up kind of relationship. It's fine because it can happen with things like nursery rhymes, I think, but we're missing a lot if we don't look at nonlinear coupling. So increasingly these days, we're looking at nonlinear relationships in brain-to-brain signals, and we use things like weighted symbolic mutual information, which is an information theoretic approach, and we can look at state changes or symbol changes in one brain versus the other, and dynamical systems theory. So I'll show you how we've instantiated that for our animal model. This is where we look at states, trajectories, and attractors. Okay, a little bit more geek work in case anyone is interested in how intricate this is. We also compared parameterization. So I'm going back to Daniel's talk on the multiverse, right? Parameterization. How does changing one setting matters immensely. So here, this was actually awarded best paper by a very geeky IEEE conference, because we were just asking the question, what is the optimal epoch length? Let's say you want to analyze EEG to compute connectivity. You have to decide what your time window is, right? Everybody argues about this. So we wondered, is there one ideal epoch length, or does it depend? A lot of people think it depends, but depends on what? So here we tested 2x2 analysis on whether you were computing a linear measure or a nonlinear measure, and whether you were computing it for the baby or for the adult. Surprise, surprise, there was no one size fits all. The most optimal epoch length was a double dissociation. It was opposite for adult and baby, and for linear and nonlinear. Why? Because there's a developmental phenomenon here. We're writing a paper which is going to propose that over development, we start off with stronger linear connectivity and we develop nonlinear connectivity as we go along. So if you're describing the baby's brain, then linear measures are probably quite good. If you're describing the adult brain, you really need to tap into the non-linearity in that. Okay, geekiness over. So that's that paper. The last thing is just to quickly introduce dynamical systems theory for you, and this is really a bit more abstract. But it's thinking about your cognition and your behavior as a state space. There are a number of different states that you can occupy at any given time, and your environment or parameters in your brain tilt you into one state or another. So here is an example of a system which is either very integrated or very segregated, and that varies as a function of the excitability or the gain of the system. You can visualize this as some kind of a landscape as well, some kind of energy landscape, and show that this system can transition from being very integrated or very segregated, depending on these things like how excitable or how much gain there is in the system. So this is a much more sophisticated way of describing the range of possible behaviors and the range of ways that we can couple with each other, because it's not just on, off, high, low. It's a 3 or even a 4D space that we can potentially occupy, and which is negotiated between the two partners. Okay, so entrainment, does this work? I'm just going to quickly introduce this because this isn't really core to today's talk, but this was a paper with an adult cohort, Cambridge undergraduates, where we tried to drive entrainment in the visual cortex. So the task was perceptual discrimination. They were given a target which could either be concentric or radial like this, and you had to see which pattern it was. But before they saw the target, we entrained the visual cortex by flickering a visual stimulus at a particular rate, and this was the key. The rate that we gave the flicker was either matched to their alpha or non-matched to their alpha. So if they had a personal alpha of 10 hertz, we would either give it on the dot 10 hertz flicker to match, or we would be off by 1 hertz. We would flicker it too slow or too fast. Okay, match versus not match. And the other thing was, after we gave the entrainment, we gave the target either at the trough or at the peak of the entrained oscillation. So we played with rate and we played with phase. And to our surprise, we got this huge effect that when you entrain the visual cortex at the right rate, match rate, and you deliver the stimulus at the right phase, trough phase, we increased learning by 3-fold. In fact, the shocking thing was these Cambridge undergraduates who sat through all 900 trials, if they got the wrong entrainment rate or phase, they failed to learn completely. This wasn't an IQ problem. Okay, it was because we were off cycle and giving the information completely at the wrong phase of the brain to capture it. So clearly there are very powerful bottom-up entrainment effects that we can tap into with some of these social cues. So although this was artificial, if you can imagine things like nursery rhymes, singing at the right rate, like I said, matching the song rate to the chance oscillations, we can harmonize those two and increase learning in a very analogous fashion. Okay, so we could show this in adults with 900 trials. No chance we can do that with a baby. And we're thinking of using this with BCI technology. In the last bit of my talk, I just want to now show you a little bit about what we've done with the animal work. So we spent some time looking at rodents, mice, mummies and babies. There is a good reason why I think they have not evolved better past their current level, because mice mummies are terrible parents. You know, they only care about themselves. They will abuse their kids and eat their kids. So I really think there's probably a good reason why the very good parenting that human species have involved have made us, be able to retain, pass knowledge, and grow and evolve a lot beyond. Having said that, there are still some commonalities in the social development. Over time, the pup develops capacities to engage with their peers. There is still licking and grooming behavior that is very similar to the way that we touch and stroke our babies. There is a social play between peers as well that's similar to the way that our kids would play with each other. And of course, there's communication. So some of the basics are still there. It's just that the rodents have really not optimized this for their development of their pups. Okay, so I'm going to end with this one example here where we did an optogenetic study. And this was to really strongly test the causal hypothesis that synchrony matters. And it's not just an epiphenomena that we're measuring that is, as by chance locked to something in the environment. So optogenetics is a technique where you can trigger activation of particular neurons in the brain at will. It's light activated. So you can trigger the activation of neurons by application of a light signal. So we created an optogenetic dyad. We had a mum and a pup who were both optogenetically mutated. And we could trigger the activation of the parvalbumin neurons. So these are PV interneurons in the prefrontal cortex at will. And we chose the PV interneurons because these drive the gamma rhythms and the gamma cycles in that part of the brain. And so we could then synchronize the mum and the baby pup perfectly by driving the light signals, so we have perfectly synchronized, or we could break the synchrony. You could desynchronize them by giving randomly patterned stimulation. And we did this in the context of a social transmission of food preference paradigm. So in the wild, mice can learn vicariously from each other about food that is safe to eat. So if one mouse goes out and it eats a different food, a new food that has never been eaten before, and they go back to the nest and they interact with other mice, the new mice will smell or interact with this, demonstrator mouse and learn vicariously about the food that they've been eating. So that if you subsequently offer the new mouse two foods, the one that they smelled and a different food, they will eat the one that they smelled more. So this is a nice example of vicarious or social learning, and our babies can also do social learning like this. So we chose this paradigm because of its analogies to human context. So just to go through that again, there was an acquisition phase where the dam, the mother, would eat one of the flavored foods, either cocoa or cinnamon, and the dam and the pup would then interact together across this wall and then in free interactions for 25 minutes. And then we tested whether the pup would acquire the food preference. And we did the optogenetic stimulation during the social interaction phase here. And we predicted that if you would synchronize them, you would change the learning characteristics and how information was transferred during this interaction period. Okay, so first of all, did we get a main effect? I'm happy to say yes. The synchronization did change the pup's subsequent learning and foraging behavior. Most specifically, it changed how much time they spent hanging around the bowl that contained the mummy's demonstrator food. And you can see the exploration raster here, that what we're seeing here is not a change in appetitive behavior, because it's not the case that they just ate more food overall. We see sustained exploration of the demo food sets in the bowl all the way towards the end of the experiment, even after the pup is full, which means they're not just learning to fulfill their hunger, they're learning a superordinate goal. Something is motivating them to hang around the mother's scent longer. So this is socially motivated foraging, not just hunger-related foraging. So that's quite nice. But here's the state-space modeling. We wanted to go a little bit further, right? They're spending longer there, but what does it tell you about the brain system that we've created in the pup? So we did some complicated modeling. If you want to ask me about it, I can try and explain it to you. But we applied thermodynamic principles here to pull out things like the free energy, the temperature of the system, the entropy of the system, and we saw this big difference in the state space. So that 4D plot there is what I'm calling the state space, the realm of possibilities that the cognition of the pup can occupy when they're synchronized or desynchronized. You can see those two planes are very separated from each other. Long story short, the synchronized pups show much more system stability and lower entropy in the foraging behavior. So the previous synchrony has stabilized the cognition and the foraging behavior, and it's carried through into the way that they then explore the world later. We also looked at the effects on the actual social interactions while they were receiving the stimulation. And we hand-coded, this is our specialty, right? Hand-coding, we hand-code even the animals. 19 taxonomies of behavior here, all kinds of sniffing, nose to nose, sniffing the abdomen, the body, the tail, the energetic, you don't know what they're sniffing, being in close proximity. So we coded a lot of behaviors, and across all of them, you can see nearly all of them show statistically significant differences for sync versus desync, that these behaviors are longer, much more prolonged during the synchronization than the desynchronization condition. And then just to drive this home a little bit, I made my students look at the qualitative aspects as well over time. So here you can see on the x-axis the actual time of the social interaction, and on the y-axis is how likely they are to be in close proximity. This is a good measure because naturally at this age, the dads will avoid the pups. They're trying to get them to be independent, so they actually run away from the pups. So the fact that the dads spend more time in close proximity is going against their own nature. And that's what we see. The synchronized dam and pup pairs are spending more time in close proximity throughout the entire stimulation phase. But most interestingly, sometime around the 10-minute mark, nearly all the dyads who are synchronized show this unique behavior where the dam will settle down into a corner, establish a nest, and gather her kids around her, and they all huddle together. And we only saw that in the synchronized condition. We never saw it in the desynchronized condition. The desynchronized position, the dam and pup just kept running around and running around independently. So there's something about the stimulation that at some point kicked in nesting and nursing behavior, which suggests maybe the oxytocin system got activated by the stimulation. So we clearly quite changed the dynamics, and we can then put some numbers to this. We saw high system stability, contingency, and then just to close the loop, we could model how the social behaviors predict the foraging behavior, and we got a very good fit for that prediction or that simulation. And we can now, have a toy model to say, all right, if you have this level of contingency, this duration of interaction, how stable is your system going to be? And now we can play with that as a model system. So this is the dynamical systems theory approach in practice. So I think I'm quite excited about this because we've shown quite conclusively a causal relationship between synchronizing two brains and some downstream social behavioral changes and how that carries through into decision-making and exploration in the pup. So I think that's really nice that we can show this link in a rodent model. So just to summarize, I've shown you what these synchrony mechanisms could be. It seems to be the case that they're sensitive to the ostensive value and the motivational value of social signals. It can be achieved through linear entrainment, but we need to think much more about nonlinear mechanisms, especially because a lot of the effects are nonlinear. And there could be long-term consequences for children's cognition, decision-making, etc. But the remaining mysteries, like I said, we still don't know whether and how this is achieved in real life, especially without that really strong entrainment drive. Because in both these success case scenarios, we had strong nursery rhymes or we just physically stimulated the brain. Right, so how do we achieve this at all in real life? And also, how does this change your development in plasticity? Can you grow into synchrony with someone? Or out of synchrony with someone? So what are the plasticity mechanisms that change your capacity to synchronize with someone? So these are really interesting questions, and I hope that people like you will also be interested in working on these. So thank you very much for your attention. I hope I didn't speak for too long, but I really appreciate you taking the time to listen. Thanks for your attention. Questions?
unfortunately we don't have another microphone. So thank you very much, Vicky, for your talk. It's very, very amazing, your line of research. We have time for questions. You can ask questions in Portuguese. We can translate. No issue.
Please translate.
Yeah. And I think I can start with our first question, if Guilherme. So here at the Institute, we are very much interested, as you may know, in psychiatric disorders, and at the heart of most psychiatric disorders, we have social interaction problems. We know this because the criteria says so, it's part of some of the criteria to diagnose people. So we have problems such as lack of social interaction, low quality of social interaction, and I've been thinking your new methods to understand the neurobiology of dyads and social interaction, what would be the implications for psychiatric disorders to go beyond of what we already know? Because we know there's some issues there, but what do you think that it can contribute in terms of causal mechanisms, or even in terms of interventions in the near future or long-term future?
It's a great question, Daniel. So partly answered this in the talk at ACOPA with the screening tools, but I think from the scientific perspective is one of the reasons why I want to include this model here. Because once you have a parameterization of the system, you can then see what happens when the values of that system become more outlying or more atypical. So here we could actually fit, I didn't get a chance to talk about this in great detail, but okay, the key things are sync versus desync, right? So synchrony was the green curve and desynchrony is the red curve. And here energy relates to the stability of a system. And the more negative the free energy, the more stable the system. So it's quite clear that the synchronized system is just much more stable than a desynchronized system. But what we could also play with is things like the contingency of the interaction. So we can fit in different values and see how that changes the state space. So this nice curve separation shows up when the contingency is generally quite high, which was the case in our actual data set. You get this nice separation. But if you have a diet where there's low contingency, maybe because of depression or because of some other disorder, maybe autism, where they can't really catch their cues, then you can see here the whole system becomes a lot less stable, whether or not you synchronize or desynchronize. So this is one of the ways of understanding how individual changes in these behavioral patterns could then influence downstream system stability and maybe have consequences for behavior. So this is one of the practical implications. You want to try and model the system in these multiple parameters to get a richer understanding, because otherwise you only think about one thing at a time, right? But like I said, it's a 4D, 5D state space, and each parameter that you change has an impact on the downstream effect. So this is one of the things I think we can try and contribute, which is understanding what happens when you break contingency, or when things are overly long or overly short. Actually, one of our social learning experiments, this was really funny because just a teaching experiment, right? We got the moms to try and demonstrate whether an object is good or bad to the child. We got a really interesting negative relationship between the duration of her utterance and the child's learning. And it was negative, which means that the more the mother talked about the object, the less effective the child learned about it. That was really interesting, and we don't know which the direction of causality was. It could either be, and most of us were suspecting this was the case, the more naggy the mother is, the more she just goes on and doesn't respond to her baby's boredom patterns, the more the baby gets turned off and then just doesn't pay attention to her anymore. Could be that. It also could be that, you know, the mother was getting the sense that the baby isn't understanding the task, so she's trying to scaffold and trying to elaborate, but that isn't really helping. But either way, there are these natural differences in the way that we try and modulate the interaction, which then have downstream consequences on behavior. And of course, in pathology, as you said, that can be tilted too far in one extreme or the other. So one of the things that this could help us do is find that Goldilocks point. So in our experiment with the modeling, we could find that Goldilocks point. That, okay, if you want the system to be the most stable without trading anything off, then you want roughly the durations to be at that 0.5 mark, because too little, too much, you tilt the system back up into instability. So this is one of the things that we could help to do, which is generate predictions about what is an optimal state to be in and how to push people back if they're too far in any direction. That's a very hypothetical idea. It does generate hypotheses for testing. The practical ideas I showed you before, the screening tools and the interventions.
Yeah, that's nice. Questions? Let me just... Okay.
Yeah, I mean, we've also had this discussion about, is synchrony always good? And I think that's a very important question to ask, because especially if your parent is sending very strong patterns which are maybe not beneficial for you to always be on the receiving end on, let's say in anxiety, right? Very strong, stressful signals. Then maybe being in a very high state of synchrony is not necessarily good for the child. So the child does need to learn to dissociate and decouple a little bit. So I think there's a lot of nuance here about understanding synchrony in mental disorders. And, like I said, where is that optimal calibration point? So I certainly don't think that it's always the case that more synchrony is better. I think you need to explore that state space and see what is the most stable and beneficial for development and cognition. But that requires a lot of data and modeling, I think, to figure out what that would be. Also, that landscape can be changed by drugs. So in the previous paper that I showed you, pharmacological agents can actually change how malleable that state space is. So it's another way of understanding effects of medication on your propensity to couple and your ability to kind of shift, be flexible. So I think there's lots of applications here, certainly from the hypothesis-driven side of things.
And one thing that comes to mind also is I'm very interested in causal pathways of how therapy works.
Yeah.
So I wonder if that kind of method could also have some interesting insights. I saw one study that showed that synchrony between skin conductance between the therapist and the patient somehow explained a little bit of the effect, but I assume it could go even beyond that.
Yes, indeed.
Very interesting too.
Yeah, indeed. So I did, before COVID hit, I did a pilot study on mindfulness and the therapeutic alliance between the client and the therapist.
Wow.
Yeah. So this was a collaboration with the Mindfulness Institute of Italy. They funded it because they were interested in mindfulness, right? But the idea was, I'm sure you guys know, the success of the treatment depends quite a bit on that therapeutic alliance.
Yeah.
Your ability to establish trust and build rapport with your client is pretty much the intangibles, right? And so the natural hypothesis was, is that measurable using brain-to-brain coupling? And then the secondary hypothesis was, people who are trained in mindfulness, they find it easy to establish synchrony and a therapeutic alliance with their client. So we measured about 20 therapist-client dyads with either high or low mindfulness before COVID hit. And we were showing some pretty promising results that both of those things looked like they might be panning out to be true. And you can see why, because all the hypotheses about the social cues, the attunement, when you are trained with mindfulness, maybe you are more sensitive to those cues, more able to attune, give and take. That maybe would help you to sustain synchrony levels with your client. So I think that's one way that we could, assess a marker of the attunement between therapist and client, which could have implications for therapeutic outcomes. Yeah, I didn't get to finish that because my collaborator in Anna Freud, post-COVID, she got promoted and then got way too busy. So we left it hanging for a bit. But if anyone else is interested, I certainly have the protocols to run that study.
Yeah, we can discuss this later. Yeah, I'm very much interested.
Thank you again, Vicky, for coming and for sharing all your fantastic research projects and program with us. Can you tell a little bit to the audience what you have been learning about the Brazilians, mother and child couples?
Yeah, definitely. Thank you, Guilherme, for reminding me of that. So obviously the key reason I'm here is on your invitation. So we collaborate on this Welcome Leap 1KD program, where as part of the amazing cohort that you've set up and been maintaining for 2.5 years, I got a chance to helicopter in my team and do dyadic data collection on a subset of your cohort, about 60 babies. But the nice thing was we can then do cross-cultural comparisons with my Singaporean babies and a bunch of babies in Cambridge, and then we can train the models to learn what's consistent across the 3 different cultures and also what's uniquely different about the different cultures. So we applied these techniques, the dyadic EEG, but we also measured dyadic signals in the speech patterns, emotion patterns, pose patterns, gaze patterns, etc., and physiology. So The data is still being analyzed, as you know, but one of our leading results, which we're going to present in a month's time, is the fact that the dyadic signal seems to generalize the best across sites. You know, you would have thought it's maybe the most volatile, or the most subjective, but when we train the machine models to predict across countries, the dyadic indices were the most robust. This is what really surprised us. So that connection between mom and child seems to be pretty a good predictor of developing executive function across all 3 countries. What differed by site was the contribution of the mother's signals. And so we could see that, say, I think it was in the Brazilian cohort, there was a much stronger contribution by your mother's signals towards predictive development of the child's cognition. In Singapore, it was a bit lower. And we thought this had maybe something to do with the temperament or the dynamic of the mother's interaction with the child. When I was watching the videos of the way that your mothers interact with your children, I was really amazed. Your mothers really get in there and they engage and they look at the child and they really drive the interaction and they really, put themselves in there. They're very expressive. That's really, really nice to see. Our Singaporean moms tend to be a little bit more instructive and, they will tell the child what to do, but it's very instrumental. It's like very concerned about the child's safety. Don't go there, don't do this. And it's much less, warm and engaging. So I think that's maybe what that is picking out about that particular dynamic of the interaction. And the other thing that was quite interesting was the child's own developing capacities. So we developed a new task for attention set shifting, which is a precursor of cognitive flexibility. And we found that your babies potentially are shifting a bit faster. They have more agile mental shifting abilities compared to our Singaporeans. And if you know anything about the cultures, I think this will not surprise you. Singaporeans are known for doing things by the book, liking the rules, lacking structure, don't really like change very much, approval processes take 10 years. And I think in Brazil, because your environment is much more fluid, there are more things you have to react to, we need to be more nimble and on our toes. So I think even just the environment or the way the inputs are coming is already tuning those circuits towards a higher or lower level of flexibility. So that was also really interesting to observe in these cohorts already. So I think this is what I mean by there's some consistencies. Like amazingly, the cross-brain signal was a really good predictor across all 3 sites, but there are unique aspects as well. So I'm very much of the view that there's no good and bad in any of this. Development is being tuned to cope with the challenges in your environment and your social context and your social norms. So obviously Singaporean babies, their brains are being tuned to cope with the rules and the restrictions and how to thrive in this very context. And same with your context here in Brazil. But it's just really interesting to see that play out with our measurements. So I think that helps us because it gives us confidence that we can hopefully develop a generalizable, globally valid screening tool, because we have some measures that translate really well, but we can also be very sensitive to how that might operate in different contexts as well. So I think that distinction is really important, and we're going to do that with your collaboration.
Questions, comments, please.
I was curious related to the developmental changes about this synchrony. And my question was like, do you have any studies with older kids, like preschoolers, and with the rodents, if the stimulation was only when the mice was a baby?
Yes.
Or you try to synchronize them like when they are a bit older?
Yes, that's a great question because we already know from the biobehavioral literature, so I studied this with the neural measurements, but actually synchrony has been measured with physiology and behavior for decades already. And we already know that actually the levels and amount of time spent in synchrony actually decreases quite rapidly over the first year. Ruth Feldman's studies are great for the groundwork on this one. So she had longitudinal studies at like 3, 7, 14 months. And we can see that at 3 months, moms and babies spend more than half of the time in affect synchrony, vocal synchrony, gaze synchrony. Just a lot of connection at the really early ages. Of course, as the child's capacities grow and they can explore the environment more independently, as they can drive their own interests more, so they have to be less scaffolded by the parent, then you can expect that there'll be a little bit more decoupling. So over the first year, you do get that drop just in the proportion of time spent in synchrony, and then that's a natural expectation. I don't think she measured the strength of the synchrony, but my guess is when it does happen, it's probably still quite strong. It's just the proportion of time that you see it happening in the child's life. Now, the interesting question is what happens, well, I think it will also hold true to preschool. For me, the interesting question is what happens when you become a teenager. Because as any parent here knows this is, when your kid is very, very young, you're their best friend, you're the hero, right? They will listen to you, attune to you. At some point, that whole dynamic flips. And the most important influence in your child's life is their other friends, some, random social media idols. And in some senses, you're pushing against them. Your best intentions don't seem to have the intended effect. So it's a very good question as to what happens developmentally in that adolescent stage, and whether it's a case of the preferential partners for synchronization, that top-down drive then changes to have the social value in a different social group and input instead. So the capacity to synchronize is still there, but who the child chooses to synchronize to does probably change over development quite a lot. Now, in our animal models, actually, we did do adolescent mice. I should have said this. They were on the brink. They were pre-weanlings. So they will be the equivalent of like 16-year-olds in our teenage years. Okay, 16-year-olds should be weaned by teenagers. But in a rodent's life, they're not completely weaned yet. They're just about starting to leave the nest. We had to choose that because any younger and they wouldn't be thermoregulating very well. And the hit stage was quite big for them to wear. So we had to work with slightly older rodents. So they were pups, but you could just call them pups. And I think, looking back, this also could explain why, we didn't have very, very strong social behaviors in the desync group as well, because naturally at that age, the parents are trying to split from their pups anyway. So if we could get smaller hit stages, to do the surgery earlier, probably would see stronger synchrony levels in the natural state without even synchronizing them. But yeah, that's a really great question. And I think to some extent, synchrony has to be intentional as well. You have to be able to choose who you synchronize with and for how long. That does become a bit more intentional as you get older. I would love to do this with teenagers, if anyone's interested.
This is a really interesting research question.
Yeah.
There are so many questions that can be answered by this approach. Definitely. Have you already tested preschoolers or school-age children? Yes.
I have 2 projects who will get there because they're longitudinal. We're starting at 2, they will end at 7. Mm-hmm. So I'll get there in 4 years. But they're not there yet.
And the idea is a free play or
So yeah, back to the paradigm, right? Yeah. Because we're very conscious that we want to get some usable data. So we always have a blend of more structured tasks that we're confident, like the A and B, we're confident we can get repeated trials, we can control it enough that it will be usable, and some which are more naturalistic, that that we'll do more exploratory analysis with. So combination. Because we use the wireless EEG, we can do 6-7 tasks per session. So we're not restricted to just 30 minutes and you're tied to the amplifier. Because it's mobile, we can have them rest, come back. So our sessions are like 2.5 hours, and we do 6-7 tasks. So we can have a range of measures, which is good for test-retest reliability. Yeah, that's one of the crucial decisions I made as a PI nearly 10 years ago. Do I want to use the system that all the other baby labs are using, the geodesic sponge system, because it's easy to wear and babies will complain, but I can only do 30 minutes of data collection? Or do I want to bite the bullet, do a much harder gel-based cap system, which means we have to learn how to cap and struggle a little bit with the process, but then we get stable, longer recordings. So I chose the hard route, unsurprisingly, and that's why it's kind of paid off, because our measurements are just cleaner and more stable. We can do many, many more of them in one session. Yes, so if anyone's doing this, that's what I recommend, by the way. It's hard work, but it's worth it. Yes?
I was curious also, you talked a lot about mother and baby diets, but have you ever studied fathers?
Yeah, I get this question almost every single talk. It tells me that I really need to do this, right? Okay, so I haven't done it myself. Not that I'm not interested, it's because I can't get the fathers into my lab. I can't seem to incentivize them. But if you ask me to predict, I would definitely predict that you would still get good synchrony between fathers and children. Obviously, they are tuned for kids as well. What might differ is the social cues and the way they create social context to maintain that synchrony. Now, other work by people like Paul Ramchandani and Ruth as well have looked at father-child relationships, and they show similar release of oxytocin during play, etc. It's just that in the old school studies, maybe our modern men are much more, released from their inhibitions. In the old school studies, the father-child relationships were a lot more restricted to kind of rough and tumble play, less affectionate, linguistic, kind of singing nursery rhymes type stuff. 10 years ago, that was the situation that it's not that there's worse or stronger synchrony, it's the type of activities that are used to promote the bonding is a little bit different. But of course, I think it's time to restudy this with the modern man. I know plenty of guys in my generation who feel absolutely no shame in singing and dancing and doing everything possible with their child. So I think we need to relook this question. Yeah, do you guys have fathers coming in for Dramina?
I think so.
Yeah, sometimes.
Oh, that's wonderful. Because your cohort is big enough that you could actually do this. Yeah. Yeah, we asked mothers just for convenient sampling. They seem to be the ones who are willing to sacrifice their weekends, etc., to come in. Yeah.
I think some of them even complained, right, that they didn't get enough questions and something like that.
Yeah.
Because it's all about the mother.
Absolutely. In fact, these days we want to try and do a triadic interaction because I think the bond between the mother and the father is also really important for stabilizing the bond with the child. So I've only been talking about two ways, but of course the family is multiple relationships, right? So we can certainly look at triadic interactions because they all influence and balance each other. That's why that state space is so powerful for showing that balance of factors. So I think that is another challenge we can take on in future. If anyone's interested. too many proposed research projects here.
The CID system has a triadic measure, right?
Yes,
yeah.
exactly. So I think it would be great to validate that with the brain signals if we could. And it's not that difficult to acquire 3 concurrent signals. In the analysis techniques, okay, you're adding one more dimension, so you just see things in an extra space, but we can do it. Yeah.
Louisa,
I saw the caps that have many channels.
Yes.
Those were simple ways to get EEG signals that have 12 it's where we do it.
That's a really good question. Yeah, again, you're asking very, very good questions. So we deliberately use a more sparse array because of our gelling system, because we have to go through every single electrode and put gel in. So if we had a 64, we would never do it in time for the baby. So 32 was the max that we could go very quickly and still complete the capping in 15 minutes for the child. Now, we could show very good measurements of the network even with that. But the truth of the matter is you don't actually need that. A lot of our measures are very spatially located. So we get very consistent measurements across the frontal regions, central regions, and parietal regions. So in principle, now that we've done a lot of these, you could do it with an even sparser array that just samples frontal, central, parietal. What I wouldn't sacrifice is the quality, because the problem with a lot of the mobile gaming unit headsets is that the dry systems sacrifice the quality of the signal. So I think we can go down in numbers, but we need the quality of the capture. And in fact, one of the things that we're doing is to work with our engineers to 3D print caps and holsters so that we can put electrodes in more strategic locations and fit them better to the child's head. Then I think you can get away with less locations. But I think you still need, for my work, you still need a good gel-based system to get those long-lasting, low-noise signals. Also because if the child is interacting, if you had a sponge, it would just fall off very, very easily. So we use that secure cap with the gel, because over time, as the gel dries out, it gets stickier and stickier and stickier. So it actually gets better. You cement the electrode down to the kid's head. So even if they're playing, the cap still stays on, the electrode stays on. That helps a lot with the noise. So these are all kind of tools of the trade if you want to try and do this and do that. But to answer your question, no, you don't need whole head. Strategic locations are good, but the quality is what you need to pay attention to. Yeah. It sounds like maybe there's interest here, and I think a great way to proceed is, if there's interest in shared supervision of PhD students, or pilot projects, I think that's a great way to go because, the more hands, the better. I can't answer all these questions myself. I think if people are interested, I'm very happy to partner to work together to answer some of these really interesting questions.
So thank you for your time. Thank you for your presentation. I hope we can continue this amazing collaboration in the future. So many ideas now, so many potential pilot projects and PhD projects. So let's discuss this in the future and continue. It will be great.
Great.
Thank you very much.
Thank you, Daniel, and thank you, Gary.